And so now we've come to what maybe we can call our third period in this afternoon. Michael W. Young is a professor at Rockefeller University. He is a biologist and a geneticist. He earned his undergraduate degree, as we heard, in biology from the University of Texas at Austin in 1971. After a summer of research with Burke Judd, and probably this emphasizes the importance of summer internships, he was working with the Drosophila genome, and that probably spurred on his interest in science because he remained at the University of Texas to complete a PhD in genetics in 1975. Michael Young continued his studies throughout his postdoctoral training at Stanford University School of Medicine with an interest in molecular genetics, where he worked in Dave Hogness's lab. From 1978, he's been on the faculty of Rockefeller University in New York. In 2004, Mike Young was appointed Vice President for Academic Affairs at Rockefeller University, and he was also granted the Richard and Jean Fisher Chair. Michael Young is a member of the U.S. National Academy of Sciences, and his research has shown how interactions among certain genes and their proteins produce molecular oscillations at the level of individual cells. I would like to ask all of you in joining me to welcome Mike Young up to the podium to deliver his Nobel lecture entitled Time Travels, a 40-year journey from Drosophilus clock mutants to human circadian disorders.
Bright lights. It's wonderful to be here. So I guess I'm the caboose. I'll pull us along and fill in a few gaps and tell you about some more recent work that's been on this remarkable journey. Let me start by just Jeff had talked about rhythms being all around us, and I think this is an example. My wife and I were out on a hike a few summers ago in New Mexico, and we came across this plant, which is called the Colorado 4 o'clock. And what I want you to notice is that all the blooms are closed. And notice this little branch here, just so you know I'm playing straight. This is at 2 o'clock when we're going out on the hike. 2 hours later, or a few hours later, we come back and we see this at 6 p.m. So this remarkable plant rhythm is believed to be due to the behavior of the pollinator, hawk moth, which is active at night in the evenings. And of course, you're in a desert environment. It's arid. Water is precious, and it takes water pumped into the petals of those leaves to open them up. So this appears to be a strategy, it seems, to conserve water in an arid environment and have those blooms available for pollination at times that make sense with regard to the behavior of your pollinator. Another example of these rhythms that are all around us is this record of hamster behavior that appeared in a symposium volume in 1960. This is each line is a 24-hour interval, so you're looking at 25 days in this animal's life in complete darkness the whole time. And it still amazes me when the animal gets on its running wheel, you get these deflections from that line, and you can predict within a couple of minutes each day when the animal's going to get on the running wheel and when it's going to get off. And you'll notice another feature of this is it's not 24 hours. It's about 24 hours. It's circadian, which, how could it be anything other than an internal reflection of some kind of an internal pacemaker? As Michael pointed out, we have still other phenotypes as we acquire new tools to look more deeply for rhythmicity. These are rhythms that are established in the head of the fruit fly. This was of Drosophila. This was work of Adam Claridge Chang and Herman Wienen about 15, 16 years ago using microarray data. And the way you read this is that this is 150 gene patterns stacked one on top of the other. Each row of red and green boxes is the pattern of a single gene followed over 6 24-hour intervals, some with a light-dark cycle, but others in complete darkness. And they're stacked according to time of day of their maximum expression. So these would be expressed maximally at dawn, at noon, these at dusk, these at midnight, and so forth. And I think what I hope you see are red and green stripes running top to bottom, just illustrating these waves of expression over this very large group of genes. And the other thing I'd like to point out is that those are not vertical stripes. They're running diagonally across the chart, which is pointing out for us that this timer is not simply swinging back and forth between two states, day and night. It's a program that generates patterns of gene expression where every hour of the day there's a unique relationship between genes that are hitting their peaks and genes that are hitting their lows. The next slide shows that same pattern, but now next to it are results that are obtained with 3 clock mutants, period, timeless, and clock. So those are null mutants, and where are the stripes? The stripes are now gone. And as Michael pointed out in his talk a few minutes ago, what we appreciate is that not only does the behavior go away with these mutants, but all of the genetic changes that are programmed, this temporal program is eliminated by loss of those genes. And this is unique to the genes that contribute to this pacemaker mechanism that we've been studying. So just to give you an overview once again of the way this pacemaker works, we have the period and timeless genes on different chromosomes in the nucleus. Transcripts are produced by association of clock and cycle, activating transcription of both per and timeless. Their RNAs are translated in the cytoplasm to give timeless and period proteins, but then we run into some post-transcriptional breaks on the system that produce the oscillations that we see. One of the important initial breaks is produced by a gene we called double time. And this turned out, when cloned and sequenced, to be casein kinase 1. And that kinase phosphorylates the period protein and marks it for destruction. So at the early stage of each daily cycle, when period proteins and timeless proteins are at very low levels, double time, which is constitutively produced at high levels at all times of day and night, is the first partner that the period protein sees. And so most of the early period proteins that are produced are immediately dispensed with in the cell. On the other hand, many hours later, as timeless proteins build up, as we have higher and higher levels of timeless proteins, we now can produce something new, which is a period timeless dimer. And period timeless dimers are immune to this phosphorylation-based destruction by double time. And they also have a property that they now seek the nucleus, whereas period and timeless on their own seek the cytoplasm. And of course, as you've already heard, period and timeless are inhibitors. They bind to physically clock and cycle and repress the action of those activators to close this cycle. in a light-dark cycle, that would all happen in constant darkness, which is why we see oscillations principally in constant darkness. But in day-night cycles, we also have the issue of these clocks following the phase of the environmental cycle, and that's achieved in the fly by a protein called cryptochrome, which is a photolyase-like molecule that absorbs light. When it absorbs light, it physically associates with timeless, recruits proteins that will cause both timeless and cryptochrome to become phosphorylated and degraded, so that all day long, when the sun is up, we have no timeless because of the activation of cryptochrome. And of course, if you have no timeless, all the period protein will only have one partner, which is double time, which means we also have no period. So we have massive amounts of RNA for both period and timeless during the daytime, but it's sterile in the sense that we're not accumulating protein until the sun goes down. And this is essentially how the phase of this clock is established. And as we move a fly from New York to Stockholm, for example, you can see the effects that would be expected from this kind of a mechanism. So as Michael pointed out, there's a great deal of homology when we move from the fly to other animals, the rest of the animal kingdom. So in us, for example, we have many familiar proteins. We have clock and BMAL, which are the orthologs of clock and cycle in the fly. We have 3 period proteins, per1, per2, and per3, all of which are related to the single period protein that we have in the fly. We also, once again, have casein kinase 1, the double-time ortholog in mammals, which once again controls the accumulation and fate of the period protein. One difference, an important difference we have is whereas cryptochrome was a partner for timeless controlling its accumulation, now cryptochrome has become the partner for per, which is an interesting evolutionary switch in dance partners. But it's a cryptochrome per dimer instead of a timeless per dimer that now moves to nuclei and inhibits the action of clock and BMAL. And I'll come back to these differences in a few moments when I begin to speak more about human clocks. So originally, the discovery that clocks were found all over the body was a discovery in flies. It was made in Drosophila by Jeff Hall in collaboration with Steve Kay in 1997. And they had generated a form of per which was connected to luciferase so that you could use a light monitoring scheme to image live Drosophila and all the intact and dissected body parts of the fly, and realized that there were per-driven cycles all over the fly. And as Michael pointed out, we now know that much the same is true for mammals, including ourselves. And in this example, I'm just giving you examples of clocks that are found in the brain versus the lung, the liver, and skeletal muscle. And I want to suggest some of the things that might happen to you if you move from New York to Stockholm, or even when you impose a brain-body conflict experimentally in the lab that can be studied continuously for many, many days. So if this mouse is kept on New York City time, it's a nocturnal animal. It's going to be active at night. It's going to be sleeping during the day if unperturbed. It will want to eat at night when it's active and be quiescent during the day. And we have these clocks in different tissues all agreeing on the time zone. They're all agreeing that they're working together on New York City time. Many years ago, Michael Medica at the University of Virginia did an experiment in which he established a brain-body conflict by allowing the mouse to eat only in the middle of the day when it should be in the middle of its sleep interval. provide food for an hour or so. After a few days, the animal learns to predict, will wake up spontaneously before the food is delivered, eat the food, go back to sleep, but then get on its running wheel and run on the wheel all night, as it would appropriately for New York City. What was found after a few days of this, though, is that the clocks, everywhere except the brain, had changed their phases by this new routine. So the brain is still stuck in New York. It's seeing that light-dark cycle. It's producing those activity rhythms that are appropriate at night. But the rest of the body is in disagreement, is not pulled along by what the brain thinks is going on. They're getting their cues directly from the feeding schedule or from other organs in the animal that are responding directly from the feeding schedule. So we have, in effect, an animal whose a collection of clocks that can't agree with one another. And this is remarkably similar to what happens when we undergo jet lag. Different organs proceed to the new time zone at different paces. We're very much, this is very much a problem of a multiplicity of clocks that can't agree for several days when we change time zones dramatically in this fashion. What we do know from these kinds of experiments is if the animal is maintained for many days or even weeks, we begin to see a number of anomalous events in metabolism and behavior that are consistent with much more severe effects that come from actually eliminating clocks in these different subsets of organs. So let me turn now to some studies that we've been doing in humans that consider a related problem, which is what if your clock doesn't agree with the environmental cycle? We've got a 24-hour environmental cycle. What if you're genetically programmed to have a clock, as we've heard in flies, like a per short or a per long? What happens if we have a clock that's out of sync with the turning of the Earth? And I'm going to be talking about a syndrome called delayed sleep phase disorder. We can think of individuals that have this disorder as night owls, extreme night owls. So they might be hammering away on their keyboards late at night, getting a big burst of energy off of their computer screen, which is, if anything, going to make things worse for them late into the night. And these are some of the attributes of these individuals. We think this appears to affect about 5% of the population from clinical studies. It's a persistent delay in the timing of the major sleep episode and strong resistance to efforts to try to manipulate sleep phase, to advance it, to make it easier to show up for work at 8 or 9 o'clock in the morning, for example. So these are records from two individuals that were in a study we began a number of years ago. On the left is a control subject, which we'll call Tau 18. And these are activity records, wake-sleep records, that involve two kinds of monitoring. One is with a wristwatch-like activity monitor, and the other is just with entries in a sleep log that's kept by the bed, so that each day or evening when the individual gets into bed, they mark the time down. And when they get out of bed, they put another mark in the log. So we have two correlated pieces of data to consider. So the way the data are plotted here is to plot not just 24 hours, but 48-hour intervals in this process. And what this allows us to do is compare the time that the individual is getting into bed with the time that the individual is getting out of bed. So you notice in the control subject from both the sleep log and the activity graphs that there's a very neat and consistent pattern in which at about 11:30 to midnight, this individual is getting in bed each night and rapidly thereafter going to sleep. And at 7 or 8 each morning, again, pretty consistently, they become active and they put a mark in the sleep log that they're getting out of bed. The DSPD subject, tau 11, has quite an erratic record by comparison, but I think the most important features are if we look at the in-bed times versus the out-of-bed times, we see that this is an individual that's always getting in bed well after midnight. And the out-of-bed times, they're becoming active again, usually just a little bit before noon. So this is a striking phase variation with regard to the timing of their wake-sleep cycle. So we were able to study that individual and several members of the family, and did a whole exome sequencing of all of these members of the extended family. And we found that there were a number of those indicated in black or gray that had an aberrant pattern of sleep, and one cousin over there with a normal sleep pattern. And then the difference that came up by whole exome sequencing in these individuals gave us one most likely candidate, which is a variation at the gene cryptochrome 1. This mutation is a single nucleotide change. It's at a spliced donor site just downstream from exon 11. And that change predicts that exon 12 will be spliced now directly to exon 10, skipping the information in exon 11. So that would predict a 72 bases missing in the messenger RNA and 24 amino acids missing from the protein. And here are we had collected dermal fibroblasts from each of the subjects in this study. There were 20 or so subjects that we studied, and you'll see subject 11 is uniquely producing 2 forms of the RNA from cryptochrome 1 and 2 forms of protein in the skin, in the dermal fibroblasts. So clearly a significant change in the structure of both the RNA and the protein in this individual. We went on to ask whether or not that modified gene was enough to change the circadian clock of a naive cultured cell. And Alina Patke in our lab did this by starting with a cultured cell that had no copy of cryptochrome. Usually would have 2 copies, cryptochrome 1 and cryptochrome 2. If you have no copies, you'll be arrhythmic. The cells will be arrhythmic. And she used this as a test system to ask, is there a difference in the pattern of circadian rhythmicity at the cellular level given a wild-type form of cryptochrome 1 or this variant form of cryptochrome 1? And as you see here, there was a very consistent pattern of rhythmicity that comes from the wild-type form, the full-length form of cryptochrome, and this truncated form gave a pattern with a longer circadian rhythm, about half an hour longer than that provided by the wild-type gene. So clearly this gene is capable of producing a change in the circadian rhythm of the recipient cells. So once again, the difference between the fly clock over here and the human clock shown on the right, important new role for cryptochrome. So this of course is the gene that is now modified in this case. Cryptochrome, the work it does is in conjunction with PER to form that inhibitory complex that suppresses the activity of clock and cycle. So keeping that in mind, Alina took a look at the interactions among these proteins. We know from that model, from what's known about the human clock and the fly clock, that period and cryptochrome will be partners to form an inhibitory complex that will then further physically interact with clock and BMAL. And so she asked the following question: does it matter with regard to the physical association among these proteins, whether you're the full-length form of this protein or the truncated form of the protein. So if I start down here, for example, and we have input proteins where we've created only the full-length protein is provided, or the truncated form of the protein is provided, or a synthetic heterozygote in which both forms of the protein are produced. What happens if you now do an immunoprecipitation with a target of hemagglutinin-associated clock? And what is found is that the full-length form of cryptochrome is very weakly recovered, much stronger recovery of the truncated form, and in this synthetic heterozygote, again, a strong bias toward the truncated form of the protein. Up here on the left is just a reciprocal experiment where the focus of immunoprecipitation is BMAL. And again, the inputs are over here, and we see much the same response. The full-length protein is recovered poorly relative to the robust recovery of the truncated protein, and the synthetic heterozygote is strongly biased toward the truncated form of the protein. In contrast, if we look at what's happening with interactions with PER itself, how do these two varieties of cryptochrome interact with PER itself? We find, here's the input, that the input and the pull downs look very much the same. So in summary, what seems to be the case is that period and cryptochrome appear to have a strong physical interaction that is not affected by this mutation, but the interaction of the period cryptochrome heterodimer with its target activator proteins, clock and BMAL, is strongly altered by this mutation. So we thought all of this was extremely interesting at the level of molecular biology, but we were really stunned when we sat down at a computer a few years ago and asked, well, how common is this mutation in human populations? And this is what we saw when we did that, looking at the exact database available through Cambridge. The overall world population, the frequency of this allele suggests that carriers, and again, this is behaving as a dominant so far as we've seen so far, carriers would be almost 1% level in the population. If we focus on Europeans, those that are European or of European descent, the allele frequency is shown here, so twice that would be the carrier frequency. That's about 1 in 75 individuals of European descent. So we were stunned that this allele, which had these features changing the function of the clock, was so prevalent, but it did suggest that we should be able to identify without a lot of trouble additional carriers to confirm this relationship. And we set up a collaboration with Tayfun Özalek at Bilkent University in Turkey, who had been working with a colleague across the hall, Jeff Friedman. And Tayfun recognized a number of families that had carriers, both homozygous and heterozygous carriers. Large families, only a few of which are shown here. And by looking at the patterns of sleep in all of these individuals, we're able to compare the patterns shown in the filled boxes, the dark boxes, carrier boxes, versus those in the unfilled boxes, which are non-carriers. And we can plot the behaviors of these two groups as shown here. The non-carriers, shown over here, that have what we call a mid-sleep measurement on free days. So on free days, when do you go to sleep? When do you wake up? And what's the midpoint of that interval? And for most of us, that's going to be around 4:30 in the morning. And that's true for most of the non-carriers in these families, but their relatives who are carriers, whether heterozygous or homozygous, have a very different pattern that's shifted by about 2 hours, very much like the original proband that we studied, where the mid-sleep point is about the time that most of us would be getting up and taking off for work. There's a second phenotype that emerged, which are fragmented sleepers. These are individuals that point to their major sleep episode as a short nap that can be early or late in the evening. Again, something not seen in the non-carriers. So to summarize what we've seen, we've got a number of unrelated families in which the presence of this variation predicts DSPD, the high frequency, the high penetrance of this mutation with regard to that behavior suggests a significant contribution to DSPD worldwide. Since the mutation is found in multiple members of each family, it's running through the family, it's being inherited, we have to assume that every tissue has these altered clocks. The variation shows enhanced binding to clock and BMAL, as I pointed out, and it appears to be a strengthened transcriptional inhibitor by other work that I don't have time to talk about today. The competitive binding to clock and BMAL suggests a molecular basis for the dominance of this trait. And finally, as I mentioned, it's sufficient to express just this one gene in individual cells in culture and recapitulate an altered circadian rhythm. So lastly, I'd just like to point at some questions we have for the future. One of the problems in studying sleep disorders has been relationships to other medical conditions. It's long been the case that anecdotally a variety of metabolic and psychiatric disorders are often said to be associated with disordered sleep and very often delayed sleep. And but it's been difficult or impossible to establish causal relationships. But what we have here, I believe, is a new opportunity. If you have large numbers of subjects that are available to study, we should be able to test whether a particular sleep mutation extends to other medical problems, and particularly when the mutation can be studied as it's traveling through unrelated families, we should be able to rule out the impact of environment and make some clear conclusions about the relationship of mutations of this sort to other medical disorders. And lastly, let me just point to the work, to the people, the AIs, as Jeff put it, in the lab that have contributed to this work over the many years. In the 1980s, Ted Bargello and Rob Jackson were involved in the initial mapping and cloning of the period gene, sequencing, identifying the nature of the mutations that Ron Konopka and Seymour Benzer had initially found some years before. In the 1990s, Leslie, Amita, Jeff, Lino, and Adrian all worked on the discovery and characterization of the timeless gene and its protein. Shortly thereafter, Jeff Price, Lino, Adrian, Justin, and Brian all worked on the discovery and characterization of double time, whose role I told you about. And in the 2000s, we began to move into the era of microarrays and looking at the larger patterns of gene expression that are controlled by these clocks, and that was principally driven by Adam Claridge Chang and Herman Veenan. But still more genes to be discovered. Sebastian Martinek found a role for GSK3, which is particularly interesting still because of its sensitivity to commonly used drug treatments such as lithium. In the 2010s, or the last 7 years, Dragan Arugulja and Nick Stavropoulos have both initiated studies of a different kind of genetics, that of control of sleep duration, the homeostatic control of sleep, which, along with circadian regulation, ultimately controls the pattern. And as I mentioned, Alina Patka has been involved in every aspect of our human work. And I'll just show one last slide, I want to point first to Lino Saez, who was a member of the laboratory for almost 30 years. He was involved in almost everything that I spoke about today. Lino unfortunately passed away in 2014, but I know he would really love to have been able to be here. Alina Patke, again, has been responsible for every aspect of the human work. She's had her hands on every part of that project, and she is here, and I just want to thank her personally. So thanks for your attention, and for the honor.
I would like to extend our gratitude to the Nobel laureates for their extensive presentations this afternoon. May I ask the laureates onto the stage for a round of applause.